All right, let's finish up our biochemistry lecture talking about proteins and nucleic acids. Proteins are one of the most abundant macromolecules found within our bodies. They are essential for body function, for functions. When I say bodies, of course, we're talking about humans because that's the most easiest to relate to. But they're essential for life. They're essential for life functions. They have a lot of different things that they can do. They can break down the food that we eat, such as amylase in our spit breaks down carbohydrates. They transport things, such as hemoglobin, which transports oxygen around the body. They're structural, like actin, which is a main part of our muscles, and keratin, which makes our hair and our nails. They're signals, they're hormone signals such as insulin that lets us to regulate our blood sugar levels. They're defensive. They're part of our immune system. They can contract. We talked about actin being part of our making up our muscles. The actin and myosin within our muscles interact and allow for contraction. And then they can store things. So things like legumes store protein. So you think legumes, they're like plants like peanuts and alfalfa, and then egg whites or albumin stores energy or stores nourishment for those developing birds, embryos, egg whites. So mainly right now we're going to focus on enzymes. Enzymes catalyze biochemical reactions, so that means they speed up those biochemical reactions. Enzymes are extremely specific. So specific enzymes are only going to break down specific molecules. Examples of types of enzymes: catabolic enzymes break down substrates, so amylase that we talked about in the last slide, break down carbohydrates. Anabolic enzymes build complex molecules. So anabolic enzymes are going to help build more complex structures, and both of these are catalytic enzymes. They affect the rate of chemical reactions. All enzymes are catalytic. They help to increase in most cases, the rate of the chemical reactions. Proteins can also be hormones, which are chemical signals. So we release hormones in our bloodstreams, and it moves to different areas of the body, and it acts as a signal so that different things can occur. Hormones are typically made up of small proteins or steroids, like the cholesterols we talked about last lecture when we were talking about lipids. Hormones are secreted by endocrine cells. The endocrine system is a slow reaction system, but it has a long-term reaction. Like we talk about our body systems that allow us to react to different things. Our nervous system is very fast acting and it's very short acting. So for instance, like when we put our hand on a hot stove and we pull that back, that's a fast reaction, doesn't last very long. Hormones in the endocrine system take longer to respond and last for longer. So think about like the onset of puberty. We don't just thankfully, or maybe unthankfully, I don't know, depends on how you look at it. We don't just hit puberty overnight. It's not an on-off switch. It takes a little while for our bodies to get those hormone levels that it needs in order to achieve that puberty. And then it will last for a long period of time. Enzymes are either globular or fibrous in shape, so that they have different shape structures, and that shape is critical to function. When we talk about enzymes, we talk about proteins being globular or fibrous and how it's critical to function. If a protein has its shape, then it should function. If a protein loses its shape, it won't function. And denaturation or denaturation is when that protein loses its shape, when it loses its function. Now, what causes denaturation? Changes in temperature, too hot, too cold, pH, too acidic, too basic, or chemical exposure. So certain chemicals can cause changes in protein shape. Proteins are made up of amino acids. There are 20 amino acids. 10 of them are considered essential because our bodies don't make them. We have to eat to get them. They have to be attained via our diet. What amino acids are essential to which organism varies, but in humans, we have to eat in order to get isoleucine, leucine, and cysteine. So we have to consume food to get these amino acids so that our bodies can build proteins and our bodies can function. The monomer for a protein, which is the polymer, is the amino acid. So amino acids are the monomers for the protein polymer. Each amino acid has the same basic structure. They all have a central carbon. They all have an amine group. They all have a carboxyl group, C double bond O, OH, right? They all have a hydrogen group. And they all have, oh, I did this one wrong, I think. There we go. And they all have an R group. And the R group is known as the side chain. And it will vary from amino acid to amino acid. The R group differs for each of these 20 amino acids. And the difference determines the nature of the amino acid. Some of these R groups make the amino acid polar. Some will make it nonpolar. Some make it acidic. Some will make it basic. All right. The amino acids are covalently bonded together. So they're going to bond with another amino acid over here to form what's called a peptide chain. Now, remember, every macromolecule is covalently bonded together, and they all have specific names depending on what we're talking about. A carbohydrate is a glycosidic linkage. A lipid is an ester bond or an ester linkage. And proteins, they are called peptide bonds. So when we bond a bunch of amino acids together, we form a peptide or a polypeptide chain, a series of amino acids together in linear form. This is called the primary or the first level of protein structure. So a polypeptide chain is literally just a chain of amino acids bound together. So this is our basic amino acid structure. So every single one of them will have an amine group. They will all have a carboxyl group. They will all have a hydrogen and a central carbon. The difference between them is going to be this R side chain. And you can see in this one all the different side chains that amino acids could have. All right, so these all make them positively charged. These side chains make them negative charged. These side chains make them nonpolar. They give them different properties. When they bond together, we're talking about a covalent bond. We're talking about dehydration synthesis. So we are removing the molecules needed to make H2O, and we are bonding them together in a peptide bond. Protein structures can be made up of multiple polypeptide chains. All right. So a protein structure is when you have that chain of amino acid folded up together. It can be made up of a single polypeptide chain. It can be made up of multiple polypeptide chain. Protein structure, shape is critical to function. If the structure doesn't have the right shape, it will not work. There are 4 structural levels: primary, which is what we just talked about; secondary, tertiary, and quaternary. Primary structure is made when the one amino acid group binds to another amino acid group. They're joined by those peptide bonds. The amino acid sequence is determined by your genes, by your DNA. All right, the DNA tells us what order to put those 20 amino acids in. The final structure is determined by the amino acids themselves. Remember, we talked about amino acids having different properties because of the R groups that are attached to them. Nonpolar, polar, charged, not charged. The interactions of those R groups determine the folds and twists in the secondary, tertiary, and sometimes quaternary levels of structure. Sometimes things go wrong and it can affect the way the protein works. Sickle cell anemia is an example of an error in primary structure. So if you remember, primary structure is just the string of amino acids. Sickle cell anemia is an inherited form of anemia. And anemia is when we have low red blood cells, low oxygen. It's characteristically low iron because iron is found in the heme groups of the amino of the red blood cells. So the red blood cells are deformed in this form of anemia. Sickle cell, they're deformed and results in a decrease in O2 available for the body. Sickle cells are rigid, they're sticky, and they're sickle shaped, or they're shaped kind of like a crescent moon. They get stuck in blood vessels. It decreases the amount of blood flow, and it will decrease the amount of overall oxygen in the bloodstream. It's caused by a single amino acid substitution. So in the case of sickle cell anemia, one amino acid is wrong. It's the result of a mutation. And just because this one amino acid is wrong, it changes the protein structure, changes the shape, and therefore changes the function. And so that results in sickle cell anemia. So you can see right here, you got a sickle cell here, you got a sickle cell here, you got a sickle cell here. And that is resulting because instead of getting our glutamic acid, it gets substituted with a valine. And it just changes. So that's saying like this one is correct. This one right here is incorrect. That's as basic as you can get when it comes to an error. Change that one protein. Change that one amino acid. It changes the entire protein, and it changes the entire structure of those red blood cells. Right. So these are normal red blood cells. They're kind of circular, right? And this dude right here is a white blood cell. So just so you can know what you're looking at here. So protein structure is extremely important. And if we lose that shape, whether we're talking about primary level shape right here or more advanced levels, we lose the function of the protein. Protein structure secondary level. So primary is a chain. Secondary, we start to see folding. That folding is either a pleated sheet, so it's bent like this, or it's called an alpha helix. A helix is that spiral shape. That's all you really need to know. In the secondary structure, you see a helix, which is twisted or corkscrewed, or pleated, which is an accordion fold or a pleat. All right. Tertiary structure is when we start to get a three-dimensional shape. This is when the side chains, the R groups, start to interact, and instead of having, our chain or our pleat or our helix, we start to see a three-dimensional shape. It is the final shape of proteins made from a single polypeptide. If our proteins are made up of multiple polypeptides, so multiple strands, you have quaternary shape. Hemoglobin is an example of a protein that is made up of multiple polypeptides. So you see 4 different polypeptides in this hemoglobin molecule. So the interaction of those subunits, when we bring those multiple proteins together, multiple polypeptides together, you form the overall protein. Hemoglobin is a globular shape. We talked about globular versus fibrous as a characteristic for proteins. It is the protein that carries oxygen and iron within our red blood cells. So to review, you have 4 levels of protein structure. Primary is just a strand of amino acids. Secondary, they'll start to pleat or twist. Tertiary, you can see see in this tertiary, what am I doing here? In the tertiary, you can actually identify, here's a pleated section, here's a twisted section, a helix section. So you can see in there start to get you a 3D shape, and in quaternary you have multiple polypeptides. All right, denaturation. When proteins lose structure, we lose the structure. When we lose its structure, we lose its function. So denaturation is different from like our sickle cell anemia, where we lost structure because we had a mutation. This, we maintain the primary sequence, but we lose the overall shape. It's caused by changes in temperature, pH, and exposure to chemicals. Often it's reversible because we keep that primary structure intact. So for instance, in your body, if it gets too hot or if it gets too acidic, let's say you're running at an extreme high fever, it gets to the point where it denatures a protein. When that fever goes away, it goes back. So typically when we return back to that homeostatic level, it can return back to its original shape. There are situations where it's irreversible. When it is damaged because it's too high or too extreme. So, an example of an irreversible denaturation is like frying an egg. The albumin in the egg, the proteins in that egg, when we cook it, we can't uncook that egg. It has gone too far. There are also examples of proteins that can handle extremes, that can handle high temperatures or extreme pHs. And so, for instance, bacteria in hot springs can handle that temperature. The digestive enzymes in our stomach can handle that pH caused by our hydrochloric acid, which is a pH of 2. All right. And I'm thankful for that bit because those enzymes can handle those extremes. I can break down my hamburger. Some proteins will receive help in folding into the correct shape by proteins called chaperones. And those chaperones help just make sure those proteins fold correctly. All right, we are at our final macromolecule with nucleic acids. Nucleic acids are essential to the continuity of life. They are essential for inheritance, and they are essential for protein function. DNA is our inherited molecule. RNA is the molecule that allows us to build proteins. DNA is deoxyribonucleic acid, DNA. It's found in all living organisms. It is the primary information molecule. It guides protein formation. It carries information from generation to generation. When we talk about the entirety of the DNA within a cell or within an organism, we're talking about the genome. And they are made up of 2 linked chains. It is a double helix. So it's made up of 2 linked chains, and it contains coded instructions that allows us to build all of the proteins that we need to function. Ribonucleic acid, RNA, is specifically involved in that protein instruction. It carries the information from the DNA out of the nucleus, transports it to the ribosomes, where it brings in amino acids and builds proteins. Nucleic acids are made of monomers called nucleotides. Those nucleotides are made up of a sugar, a phosphate, and a nitrogenous base. All right, so this is a sugar, a phosphate, and then a nitrogenous base. So this subunit right here will bind to another subunit, right? The slide's starting to get messy. And form one side of our nucleic acid. So nitrogenous base, a sugar, and a phosphate. They are covalently bonded together, and the bond for nucleic acids is called a it's not erasing, erase, a phosphodiester linkage. All right, so let's just think about this. Let's talk about what we're looking at here. Covalent bonds for macromolecules. Not right now. Covalent in macromolecules. Carbohydrates, glycosidic. Lipids, ester. Proteins, peptide. Nucleic acids, phosphodiester. All right. All right. Nucleotides have nitrogenous bases. So remember, we got a phosphate, we have a sugar, and we have a nitrogenous base. And that nitrogenous base is different depending on whether you're talking about DNA versus RNA. In DNA, we have adenine, thymine, cytosine, and guanine. In RNA, adenine, uracil, cytosine, and guanine. We don't have thymine in RNA. Purines are adenine and guanine. They are 2-carbon nitrogen rings. So if I was to draw a purine right here, I would draw it so it's got a double ring. Right? A pyrimidine would be a single carbon nitrogen ring. So we'd have a phosphate, a sugar, 5-carbon sugar, and then we would have a single nitrogen ring. Cytosine, thymine, and uracil. The 5-carbon sugar, pentose, pent means 5-carbon sugar. In DNA, that 5-carbon sugar is called deoxyribose, and then in RNA, it's called ribose. So if we look at our diagrams here, we have a carbon here, a carbon here, a carbon here, here, and here. 5 carbons. Right. When we bind nucleic acids together, the phosphate of one nucleic acid is going to bond to the hydroxyl group of the 5-carbon of the sugar on another group. Right? And that's our phosphodiester linkage. So literally, let's do orange here. This phosphate right here is going to bind right here. And that's going to join them together. Right? DNA is double-stranded because you got 2 strands there. RNA is single-stranded, so you just have the 1 strand with RNA. Our nitrogenous bases, so this is our phosphate, our sugar, and our nitrogenous base bound to a phosphate, our sugar, and a nitrogenous base of another one. On DNA, you have 1 strand going this way and 1 strand going this way, and both strands are covalent, are connected with hydrogen bonds. Right? See, DNA is double helix, means there are 2 strands. They have a sugar and phosphate backbone. Right? So when we look at the sugar and phosphate backbone, I'm going to highlight that right there. So this is our sugar and phosphate backbone. It's made of our phosphate group and our 5-carbon sugar. And then the nitrogenous bases are paired. Let's do a different color. On the interior links, that should be links. Links. Or the rungs of the ladder. So we highlight that. These are going to be our nitrogenous bases right here. All right. The 2 strands of DNA run in opposite directions, So that means that this one is going this way, and this one is going this way. It's called anti-parallel. So they're still parallel, but they're opposite of each other. And then we have our nitrogenous bases are paired together. Adenine will only pair with thymine, and cytosine only pairs with guanine. Right? So if I have only half a strand of DNA, I have adenine, thymine, adenine, cytosine, guanine, adenine, right? I can make another strand of DNA using that pattern. So adenine pairs with thymine always. So I've got thymine here, I'm going to have an adenine here. If I have an adenine here, I'm going to have a thymine here. If I have a cytosine, that means I'm going to have a guanine. If I have a guanine, that means I'm going to have a cytosine. Adenine means I have a thymine. So they're complementary to each other. DNA is its own pattern. It's one of the reasons why it's such a stable molecule, and it can go from generation to generation, and it can be recreated over and over. And whenever we need new cells, we have to copy DNA. Whenever we have offspring, our DNA is passed on to them. And as they grow and they develop new cells, their DNA is copied over and over and over and over. It's a pretty efficient process. Surprisingly, not a lot of mutations, but when you think about the size of the DNA molecule, the fact that we can copy it as well as we do is an amazing fact. RNA is usually involved in protein synthesis. It is single-stranded, so it's going to just have the one side. The nucleotides for RNA are made up of very similar. It's going to have a phosphate. It has a 5-carbon sugar. It has a different 5-carbon sugar than DNA, and then it has a nitrogenous base. So this sugar right here is ribose sugar. 4 main types. Messenger RNA is created from DNA, and it carries that DNA message to the ribosome. Ribosomal RNA makes ribosomes. Transfer RNA brings amino acids into place so that we can make proteins. And then microRNA is the smallest kind, and it helps to turn on and turn off genes. So for instance, if I am a nose hair cell, I have certain genes turned on, and they are going to be different genes than the cells that make up my skeletal muscles. So gene expression is just a matter of which genes are turned on and which genes are turned off. So, DNA carries genetic information. It always stays in the nucleus. It is double-sided and twisted. So, double-sided and twisted. It has the 5-carbon sugar deoxyribose, and it is made of cytosine, thymine, adenine, and guanine. RNA is important for making proteins. It does leave the nucleus. It goes to the cytoplasm, goes to the ribosomes. It's typically single-stranded. It has a 5-carbon sugar of ribose. It's made of cytosine, uracil, adenine, and guanine. All right, so that is the last of our biochemistry lecture. We've gone through all 4 macromolecules. So as always, if you have any questions, just give me a holler, post on the discussion boards, or shoot me an email.